previous class we have studied about the photosynthesis, the different experiments regarding photosynthesis, then light pigments, let's see, that is light harvesting system, different types of pigment system, that is pigment system 1, 2. Now in this video we'll study about photophosphorylation, then different types of photophosphorylation, then electron transport system. Now photophosphorylation. What is photophosphorylation? Phosphorylation means formation or synthesis of ATP from ADP and inorganic phosphate. So the phenomena of synthesis of ATP is known as phosphorylation, and because it occurs in presence of light, that's why it is known as photophosphorylation. Now this photophosphorylation takes place in the process of photosynthesis, which is of two types, that is cyclic as well as non-cyclic. Now cyclic photophosphorylation, as we have discussed in previous class, that is there are two types of pigment system. Pigment system 1, which is also known as P700, because it absorbs, the reaction center absorbs the light, that is wavelength is P700 nm, that's why it is known as P700, and PS2 absorbs at the peak, the maximum, that is 680 nm, that's why it is known as P680 or PS2. Now this PS1 or pigment system 1, when it absorbs the light from the sun, the electrons of this orbit of this pigment system becomes excited and they emitted outside. Now these electrons are captured by the primary electron acceptor, generally plastoquinone, and it becomes reduced. Now it will give the electron to other pigment, the element of this electron transport system, and that is other electron acceptor, and it becomes oxidized. Then in this way the electron will move through this cytochrome B, F, A3, etc., and finally the electron will come to the pigment system 1, that is P700. During this movement, that is transportation of electron, the energy is released which will synthesize ATP. Now from this ATP and inorganic phosphate, the ATP synthesis will take place, and that is known as photophosphorylation. So in cyclic photophosphorylation, the product is ATP, whereas only one pigment system, that is pigment system 1, is involved, and the movement of this electron is known as electron transport system or ETS. Now non-cyclic photophosphorylation. In non-cyclic photophosphorylation, here both of this pigment system is involved. Now it will start with pigment system 2. Sunlight is absorbed by pigment system 2, that is P600. Electron is emitted. Then it will be accepted by the this electron acceptor, primary electron acceptor, that is plastoquinone. Then it will move to next acceptor, cytochrome B, A3. In this way, each and every time the electron, when it is received, then it becomes reduced, and when it is released, then it will become oxidized, and finally it will come to the next pigment system, that is pigment system 1 or P700. Again now when the protein light is, the electron is moving to the primary electron system, that is known as uphill, and when it is coming down, that is downhill. Now this from this pigment system 1, again electron is released and is accepted by, here this time it is accepted by NADP, and NADP get reduced to NADPH, and this also will through the downhill, that's why it will occupy the shape of Z, that's why it is also known as Z scheme. Now at the same time here, because electron is moving from this pigment system to different electron acceptors, that's why it is known as electron transport system. As well as here one more important phenomena occur, that is splitting of water. This splitting of water, that means H2O will split into oxygen as well as proton, that is H plus, and here the product will be oxygen, and this is the electron which is coming out is compensated by this phenomena, that is splitting of water. This was not occurred in the cyclic photophosphorylation. You can also see during electron transportation from ADP and inorganic phosphate, ATP will produce, that is process of photophosphorylation will take place. So the important phenomena is photophosphorylation, that is product is ATP, then O2, splitting of water, as well as the reduced NADPH. So these two, ATP and NADPH, becomes the raw material for the dark reaction. It will be utilized by dark reaction. So it will move to the Kelvin cycle or dark reaction, that we'll discuss in next class. Now here, during this electron transportation system, the electron will move uphill, then downhill, uphill, down. That's why it is known as jet scheme. Now we can compare this cyclic and non-cyclic photophosphorylation. Only one pigment system is involved, whereas both the pigment system are involved. Here the electron is coming back to the pigment system, that's why it is known as cyclic. But here we can see that electron is not coming again back to this pigment system, that's why it is known as non-cyclic. So here the product is only ATP, here ATP as well as NADPH, and splitting of water takes place. Now there are in stroma lamellae, the pigment system 1 is present only, no pigment system 2 or the enzyme NADP reductase is there. That's why the PS1 only is there. That's why here only the cyclic photophosphorylation will take place. Whereas in grana lamellae, both the pigment systems are available, that is PS1 and PS2. That's why non-cyclic photophosphorylation will take place. In this way, we have discussed about the cyclic photophosphorylation, then non-cyclic photophosphorylation, jet scheme, then electron transport system. Now we will discuss this through the diagram.
This slide is showing the process of photophosphorylation. Photophosphorylation is the process in which the ATP is synthesized by using sunlight, and hence it is known as photophosphorylation. And here by the proton gradient the ATP is synthesized. In this process, this process is light dependent, that means the proton gradient will be created by using light, and that's why it is known as photophosphorylation, which is of two types, that is cyclic photophosphorylation and the non-cyclic photophosphorylation. This slide is showing cyclic photophosphorylation as well as non-cyclic photophosphorylation. This figure shows the cyclic photophosphorylation. Here in this we can see the sunlight is trapped by the reaction center chlorophyll P700, and it become excited and the electron is emitted. This is accepted by the primary electron acceptor, and then it will move through the electron transport system, consists of different cytochromes, and finally the electron will come back to this reaction center P700. And here during this electron transport system, ATP is synthesized by using ADP and inorganic phosphate, that is known as photophosphorylation, because this electron is coming back here to the pigment system, and that's why it is known as cyclic photophosphorylation, and output is only ATP. Now this figure is showing the non-cyclic photophosphorylation, in which light energy is trapped by the reaction center P680, that is pigment system 2, electron is emitted, accepted by this primary acceptor, then it will move through this plastoquinone, then cytochrome complex, then plastocyanine, and then it will come to the next pigment system, that is P700 or pigment system 1. Now here again it is trapping the light energy and electron is emitted and accepted by the primary acceptor. This again will be move through this electron transport chain, and it will, that is ferrodoxine, and it will synthesize NADPH as well as ATP is synthesized during this electron transport system chain. In this way, there are two output, that is ATP and NADPH, and here during the capturing of this light energy by this pigment system 2, splitting of water takes place, and thus protons are released and oxygen is released. This oxygen will be released in this process of photosynthesis. Here this diagram is showing the chloroplast, in which the light is trapped by this grana. So, in grana light reaction takes place, and in stroma this Kelvin cycle or dark reaction takes place. Cyclic photophosphorylation, in which only one pigment system, that is pigment system 1 is involved, that is reaction center P700, whereas in non-cyclic photophosphorylation both the pigment system are involved, that is P680 as well as P700. Now, here in cyclic photophosphorylation, the electrons released are cycled back, come back to the reaction center, and that is why known as cyclic photophosphorylation. But in non-cyclic, this electron is not come back to the pigment system, that is reaction center. This in this cyclic photophosphorylation, the process of photolysis does not occur, whereas in non-cyclic photophosphorylation the photolysis of water takes place. In cyclic photophosphorylation only ATP is synthesized, but in non-cyclic ATP as well as NADPH is synthesized. Photophosphorylation takes place at two places, whereas here in photophosphorylation in non-cyclic one only at one place. This cyclic photophosphorylation not require any external electron donor, whereas here it requires electron donor either in the form of H2O or H2S. It is this cyclic photophosphorylation is not sensitive to dichlorodimethylurea, whereas this non-cyclic photophosphorylation in is inhibited by the electron flow, because it is sensitive to this DCMI, that is dichlorodimethyl